Welcome to the July meeting of Geology Club. We've got some announcements. I've got my Susquehanna Rock Fight Dirtman report. We're going to do the raffle, and then we're going to get right to Dr. Jennifer Peteya, reconstructing the color of extinct animals, from their fossils. I think it's just stimulating. So that's going to be really cool. So thank you for being here, Dr. Peteya. Jen. All right. So t-shirts, everyone loves t-shirts. Get your t-shirt now. You can take your pick from the current vugs that we have going on. We have the Maryland Titans, we have the Pennsylvania Rogues, and the of course, Geology Club standard issue in the center there. You can pick your colors, all that. So get yours now. I sent out the link is in the email that I sent out. So check that out. And here we go. Wait a minute. Yeah, that jumped. Here we go. So field trips upcoming. CK Williams, we're going to do that. I'm not sure if this one's closed out or not, but if it is, we might be running more of these. I'm not sure. We got to talk to Jake. But I am definitely looking forward to this one. This is kind of a radioactive site. It's, not going to nuke you or anything, but some really cool stuff there. So looking forward to that. And you all know Professor Steve Lindbergh. We went out to the new Paris quarry with him. He is helping to help get us into the U.S. Silica Berkeley Springs facility. This is a plant that I've always wanted to get into. But right now, the middle of summer, it's gonna cook us. I mean, it's an oven. It's bright white sand because it's a silica plant, but there are quartz crystals and really good stuff in there. So we're gonna wait till September on that, and we should be able to get in there on a Saturday. So Professor Lindbergh is setting that up for us. And the big announcement. All right, so this is what we were discussing prior to the meeting here. This is the big trip, the New York Herkimer Diamond trip. I've pulled a lot of strings on this one. Andy was able to get with Diamond Mountain. He got us 20 free members in on Saturday. So we had put out the sign up on Friday. We have had 50 people or, 30-ish sign up for 50 people, as family members and stuff. So what we're going to do is we're going to hold a lottery drawing to select those 20 members. After that, look at this deal that I have secured for us, guys. This 50 people or $50 a person for one day. But if you're there both days at Mohawk Valley Mineral Mining, it's $80 a person for both, for both days. This is $40 a day is over half off the normal price of this place. Not to mention, it's going to be a great time to hang out, just a social kind of thing. The field after hours at night at Mohawk, I'm usually up till about 2-3 a.m. So it's just a great time around the fire, sharing stories, just a wonderful time. So I can't say enough about, Mohawk's going to be great. So those who don't get in on the 20 member free trip for Diamond Mountain, there is also, the free camping at Mohawk. So if you do get in on that trip, you're allowed to camp at Mohawk. Come this Sunday, come dig with us. $50 a person. But, so great trip. We're going to do this drawing tonight so that everyone sees it. It's fair. And this will get posted to YouTube tomorrow, so everyone will get to see that. I'm going to run through a couple more announcements, and we'll do that with the raffle announcement. So, all right, our next upcoming event, Geology Day, Indian Steps Museum. This is something I've been working on for a couple months with them. They're very excited about it. They're going to, have some cool stuff going on. But basically, they had contacted me initially to do an UV presentation and just come out and just do one day. I was like, well, let's bring the club in. Let's make it a social event. Let's do some cool stuff. You've got a museum full of rocks. So everyone come on out, bring a dish. This is going to be a potluck lunch. This is going to be kind of like the Geology Club picnic. So let's just do some cooking out. I'll bring a grill or something and bring a dish for the potluck. We'll hang out. I'm gonna do 11 a.m. I'll do a fluorescence presentation that'll leave me free for lunch to cook up some whatever, and then we'll do another one I think at 1. And then there'll be events throughout the day. I've already made 115 trilobites. So come decorate a trilobite. I've got all kinds of decorations and all kinds of cool things for that. And they provided by the museum, grinding minerals into pigments. So they're going to have minerals that you can actually grind and make your own pigments for face painting and stuff like that. That's really cool. Geology Day, Indian Steps Museum. So I hope you all come out. It's York County. It's a great location right on the river. Picturesque. There's a waterfall that we can hike to. Can't say enough of good things about this. So come on out. That's next weekend, not this weekend, but next weekend. So gonna be awesome. All right, friends of Geology Club, Jerry Jones, Zoom Rock Room on the 1st and 3rd Tuesdays of the month, while everyone's here on the 2nd Tuesday. Register for his Zoom Rock Rooms at jonesgeo at comcast.net. And they're coming up on the 150th episode, which is pretty cool. So I've been in there since like episode 6, I think 5 or 6. I don't know. It's been years. And I there's a special tribute to Jerry tonight. So Geology 365, it's where our website came from. Basically, Brian Klockner, he's a member of the club. He is the man. He gave us geologyclub.org. That's our website. So, all that. Thank you very much to him. So check out Geology 365 for tools that you can use for your own collection. All right. Area 51 miners. Congratulations on opening Area 53. They have a few sites. This one, I know Crystal Collector is actually up there digging with him right now, with Shane, the owner, right now. They're pulling some stuff out. It's pretty cool. So check out their live auctions on Instagram. That's usually Friday nights. So great deals. I've actually bought a bunch of stuff that wasn't Herkimer's. There were other stuff from his Shane's collection from around the East Coast, but great stuff that they put up on those auctions. So all right, Jeannie Smith, she's going to be here, I think it's September, for her presentation on some of the sites up in Herkimer. Depending on what happens next month with the presentation next month, we had someone cancel. So I've been searching left and right, up and down. So if anyone knows, please contact me if they want to do a presentation. But as of right now, I may do a presentation myself on Diamond Mountain and Mohawk Valley, just so you all know what to expect when you all get there. So moving on, Robert Beard, my man Bob. Check out his books. They're great resources. Even if you can't go to a lot of the sites these days, but they're still in there. There's stuff that you can really learn in there. So I highly recommend Rock County, Pennsylvania. I'm in it. So all right. Well, let's get to my Dirtman report. We'll do this real quick. So this is my tribute to Jerry Jones. He has taught me a lot over the years. Everything in this report, pretty much, not all of it, but he taught me most of it. So I just wanted to share this. Real quick, Andy, can you mute? It's bouncing back at us, just for the Dirtman report. So I actually filmed this July 4th, this past Friday, and I got it all together this weekend. So quick in the bag, got it edited, music, sound effects, all that stuff, graphics. But I really filmed this for Maryland Public Television. I'm going to be filming with them this weekend. And it actually turns out I might not be able to film in Lancaster with them, but oh well. You get to see a Dirtman report because of it, because I wanted to go out and make sure the sites were still open and available and you could see everything I wanted to see. So here we go, everybody. This is Susquehanna's Rock Fight. I hope you all enjoy. Here we go.
The Susquehanna River is one of the world's 5 oldest rivers. It has an amazing history of fighting rocks and winning. That fight has benefited humans throughout the years. Durman here, along the mighty Susquehanna River, formed over 300 million years ago. But, as a world-famous geologist once told me, it might be a bit older than that. The theory goes that the river formed initially flowing north out of the easterly lying mountains that no longer exist. However, as continents collided and Pangaea came together, the Appalachian Mountains were uplifted, reversing the flow, but the Susquehanna stayed strong and successfully won its first fight with the rocks. Long before the river started flowing, about 540 million years ago, south of the equator in warm, shallow seas, these ripple marks were left and now have been fossilized and included in Chickee's Rock. This quartzite has survived not only the coming together, but the breakup of Pangaea. And it's also moved from the southern hemisphere all the way into the northern hemisphere. And it survived. However, the Susquehanna River still cut through it, creating the beautiful, picturesque attraction that trail goers see riding by every single day. One thing enjoyed by rock enthusiasts of the different type, the rock climbing type, is this face here on Chickee's Rock. Check this out. How you guys doing?
Doing good.
What's your name, sir?
I'm Jack.
Jack? Who's that up there?
That's Brennan up there.
Brennan. Oh, wow. And they are climbing the straight vertical face of Chickee's Rock. Is he going to make it to the top? Or?
Going up to the anchors up there and to the left a little bit, up this crack down the
Okay.
So what we're doing right now is trad climbing. It's a lot of fun. We started doing it a couple years ago. It's
Okay,
flat.
so this is a popular site for this kind of thing.
I would say so, yeah.
Oh, wow. Okay, very cool. Very cool use of Chickee's Rock, something people are enjoying throughout history. Later in the Jurassic, as Pangaea started ripping apart, igneous diabase came up and filled the fractures and cracks as North America separated from Africa. Then 12,000 years ago, as we were coming out of the last ice age, major glacial outflows came down the Susquehanna Basin, cutting into the diabase, what we call potholes. Look at this little one right here. It's about the size of a finger. However, check this other one out. That's a little bigger. These potholes were created when a rock gets caught in a vortex in the water, spinning around, cutting into these rocks, leaving a smooth surface, which is great for fishermen when they're submerged by water. But when they're above water, they're great for potential gold. Man has used diabase throughout history as a super strong building material. Here at the Marietta Riverfront Park, they used it in this archway for the railroad going over this road. It's a very sturdy material. This is absolutely amazing. I'm on the river here. Check this out. There is a trapped catfish in the river. I'm about to save it. Come on. Look at that. Oh yeah. Successfully just saved this catfish. Oh yeah, here we go. Letting it go. Here it goes. That was awesome. Conowaga potholes, saving catfish. Oh yeah. But then man steps into the picture and tries to harness the river, building canals to avoid rapids and building dams to harness the energy of the river. Most early attempts failed, but modern man has successfully tamed the river so far. Has the Susquehanna officially given up its fight?
Durman here. I'm going across the Susquehanna. This is the Conowaga Dam. Look at this, everybody. Susquehanna is coming up over the dam. Wow. This is insane. Yeah, that was huge.
Wow! Durman here, bringing the geology to you from the banks of the mighty Susquehanna River. Back to you, Jerry.
All right. Well, I hope you all enjoyed that. That was filmed in about 4 hours. Did the edit Saturday and did the audio in between about 8 hours of recording on Monday and Tuesday. Got it all done for you all tonight. So, yeah, it was crazy to run into those people climbing Chickies Rock. I was at the other end of the rock filming the the ripple marks, and I come around to the other end and they're climbing the face, and I was like, oh. And then of course the catfish was like, holy crap. So I know if that was a channel cat, it was probably illegal for me to release it again, but I didn't want to kill it. I wanted to save the fish. So anyway, all right, let's keep it going here. We are going to move on to, I believe, yeah, we're doing the raffle next. Yes. Okay. Sorry. Okay. So my apologies. I just needed to get back to that. Raffle this week or this month is donated items that were given to me at one of the last field trips, and I cannot remember who gave them to me. I got a bag and I was like, all right, we're gonna give these away. Here's some wave light. It's kind of hard to see. That is wave light on that, so that'll go in the bottom. And these are actually some really decent crystals, I believe, coming from Parksburg. And then this is probably Lancaster. That is some really nice crystals right there. We've got some smoky. That's either Parksburg or Lancaster. I'm not sure. We got a little guy here. And this one is actually interesting. It's got some weird faces. It's got 4 faces instead of more. So not sure. It's just a deformed quartz crystal, but I had to think about that one for a minute. And there's a cluster. And then I donated, this is some iron stained quartz, but it's a really nice little cluster that I kind of liked. I think that's from Pickering. And Tina Kruger and I were collecting slag at the Susan Ann furnace. It's a lost history furnace that people know by a different name called Manor Furnace. And I rewrote the history of the furnace in York County because it got lost to errors since the 1800s. And I tracked them all down to the original handwritten documents, and I rewrote the history of Susan Ann Furnace. So this is 1836-ish is when this came out of that furnace. There are blues and greens and all kinds of stuff through here. So that's going in the box as well. So all of that is the raffle tonight. So everyone put in a number between 1 and 100 into the comments. And we're going to draw that after Dr. Peteya's presentation. But up now first, before we get to you, Dr. Jen, I'm sorry. We do need to take care of one thing. We're going to do a lottery for our New York trip. We want this to be fair for everyone. So I'm actually going to share, I'm not going to share the screen for that, but I'm going to share the screen for 1 second. This will be a Google random number generator. And we're going to pick 20 people to go on the Diamond Mountain trip. So first up, and I'm going to compare that to a spreadsheet that I have. You all are seeing this. So my spreadsheet goes from 2 to 29. That represents about 50 people. So each person that signed up is between that 2 and 29, but there were 2 families of 4 that signed up, all that. But this represents the people that signed up. So we will fill those 20 slots as they are generated by this random generator. And I need to be able to mark that down. I'm grabbing a piece of paper real quick. Sorry, everybody. I'm gonna write it on my phone. That's what I'm gonna do.
Copy paper. Like,
Sorry. Yes, that'll work. I got it. Cool. All right. So here we go, everybody. This is to pick the 20 people. Here we go. Here's the first generation. Our first number is 17, and I'm going to announce who that is. So I'm not sure if you're actually seeing this. Bill Geary is our first winner. Oh my God. I was going to have him lead that trip in the first place because he is the man as far as digging. Holy crap. That's cool. All right. Hold on. Everything's in the way. Go away. Sorry. I couldn't get back to my tab. All right, here we go. Okay, so the next winner of the 20 is going to be number 7. All right, and all right, this is really getting on my nerves. There we go. Number 7 is Kelly McKinty. You are on this, and Angela Dix. You are on. Congratulations, Kelly. This is why we're doing this on the meeting. You are number 2 and 3. Sorry, just writing all this down. 1 and then 2. All right, cool. Yeah, here you go. All right, next number generated is 17. We already picked 17. What the heck is going on here? 22. This is supposed to be a random number generator, and it just picked the same numbers twice. Okay, Google. Great. 22 is Rebecca Malone. Congratulations, Rebecca Malone. You're good for 1. So we are up to 4 people. Next number, you good? Is number 16, who is Teresa Conroy. You are the next number. So we're up to 5. Yep. Next number is 5. That is Mary Schreffler. So we're up to 6. I don't know what you all are seeing. Hopefully you're seeing my windows here. Yep, just 1. 7. We already had 7. 10. Number 10. Cannot believe the random generator is pulling up the same number a bunch of times. All right, so number 10 is a 4-person family. That is Dave, Tierra, Miles, and Chance Martin, I think, is their last name. But I will notify them. So that is 4 people. All right, let's keep going here. Number 15. And that is Kristen Thiel. And that's 1 person. We're up to 1, that's 3, 4, 5, 6. That's 10, 11. So we're up to 11 people. Keep going. 17 again. Wow. 10 again. What is up with this? 21. I am not relying on Google anymore. I'm going to start picking a different site. Grace, no, hold on. Yeah, Grace Moran, and Kendra Celery. So that's 2. Congratulations. Oh, sorry. Did you need that? Kendra Celery. I don't know if I said that wrong in the first place. All right, here we go. Number 11. That is Jessica Santos and Robert Quaid. So Bob Quaid will be going on this. Very cool. So that's 2.
What number was it at?
Oh, number 11. 3. So we're up to, that's 11, 12, 13, 14, 15. Jessica Santos. So we're up to 15 with that. Hit this one more time. Again. 18. Melanie Mantegna, I think. 16. So we got 4 left. We already got 15. We already got 15.
Yeah, we already picked 15. Yep.
28. Finally, something in the 20s. Jeez. Wow. All right. I am not happy with Google. Tina Kruger. Tina got it. She's going to be good for pictures. That's why I was hoping she would get. She'll take a lot of pictures. So now we're up to, what's that, 18? Yeah. Here we go. Next fire, 20. We haven't had 20 yet. 20 is Kylene Beckett. Kylene Beckett at 20. All right, so I think we have like 2 or 3 left. I'm going to hit it again. Bill keeps on coming up every single time I hit this thing. We've already had 22. 14. We haven't had 14 yet. All right, 14. Jennifer Dorrell. All right, and I think we have 1 left. And then we're going to start doing alternates. So this is 6. We haven't had 6 yet. Karen Falls. Actually, that's good that she's in with you all as number 20 because she's been there. I know she's been there, so she'll be able to tell you where to go. She's actually been in the unreleased Dirtman report that has never been released. It's the only one I've never released. Y'all good? Sorry, Corey Rockhound is doing the bookkeeping here. All right, so that is 20. All right, so we were at 20. We're gonna do a second sheet. This is gonna be our backups. So if you don't show up, or if you need to let us know, that's gonna be right through. If you let us know that you can't attend, these people will get to go. So first number for the backup. 5. 21. Those are all numbers we've already had. 27. Let's see who 27 is. 27 is Bob and Missy Turok. Good, they're on the waiting list. I've already cleared them to have a four-wheeler at Mohawk. All right. 13. Rich Casey. Congratulations, Rich. You're on the waiting list.
Question.
Yeah, first 10. So we got 3 of them now. 18. We've had 18. We've had 22. 2. TJ, Nikki, and Cadence Barnett. So that's 3. So we'll just say Nikki Barnett because I think that's the email. So we're up to what, 6? Next, 4. We haven't had 4 yet. 4 is Gunnar Kuhn. I know he's on tonight. You were on the waiting list, sir. All right, a couple more. Number 9. Michelle and Nora Bodery. So we need one more person. We've had all these numbers. 19. 19 is 3 people. Well, they got lucky. They're at the very end of the list. Miranda Luden, Tad Luden, Nikki Shaver. 3 people. That's number
You
19.
said Chad or Tad?
Tad. All right. So that wraps that up. We're good. I know.
What was the last name?
I got the number.
All right.
The sheet's not going to change.
Gotcha.
Cool. All right. Thank you, everyone. I just wanted to make sure that was fair for everyone. We had an incredible amount of sign up, short amount of time. So I wanted everyone to be fair. And like I said, amazing deal at Mohawk. If you want to still come to New York, hang out with me and Corey. It's going to be a great time. I'm telling you. Let's get to our main presentation. Dr. Jennifer Peteya. I first met her in a mass of people. I'm presenting fossils, and all the kids are running past me to her because she's got dinosaurs, dinosaur footprints. So, yeah, Gunnar was there actually that day too, and they were running past us. So, welcome, Dr. Jennifer Peteya. Take it away.
Sorry. Anyway, hello everybody. So I'm Dr. Jennifer Peteya. Sharing is not turned on. Send a request. So I am a new professor at Bloomsburg University. And I'm in the biology department, but my background is mostly paleontology, geology. I'm actually a member of Geology Club. I just haven't been able to be here for a while because of Pennsylvania Master Naturalist training. So apologies for that. All right. Can everybody, if I can get this to work, can everybody see my screen?
Yep, we got you. We got you.
Cool. Thank you. All right, now I gotta remember how to get rid of this stuff. Okay. So my background is mainly geology, although my PhD is in biological sciences, but I study mostly weird things that preserve in the fossil record, including some of the things that I'm going to talk about today. So I've studied trilobites, I've studied dinosaurs, I've studied dinosaurs, etc. So we're going to talk about reconstructing the colors of extinct animals based off of their fossils, which is what I did for my PhD research. And I apologize in advance. I have 4 cats. They're going to drive me nuts while I do this. All right. So we know, based on just visual observation, that the world today is very, very colorful. We've got some beautiful birds out there. Yeah, we don't typically think of mallards necessarily as beautiful, but look at that green on the male's head. That is just gorgeous. And these violet, purple, blue spangles on their wings as well. So birds show off a lot of amazing colors. Cephalopods, other invertebrates that are marine show off a lot of amazing colors. We've got sea urchins down here that are purple. We've got frogs that come in various colors. We've got reptiles that come in various colors. So lots of amazing diversity of color out there. And what we kind of wonder is, was this diversity the same for extinct animals? And chances are it probably was. So we're going to talk a little bit about the evidence for that. First of all, I have to talk a little bit about color and how color is actually made and produced by animals specifically. So most colors are produced by two main mechanisms in the natural world, the first of which we're probably most familiar with. Those are the pigments. So you have pigments in your body. Melanin is the main pigment that I'm going to discuss. You have melanin in your hair, in your skin, in your eyes, in your liver of all weird places. Even though you can't see your liver, hopefully it's on the inside. But we produce pigments, and so do other animals. So what pigments do is they absorb certain wavelengths of light and reflect others. In this case, this is a northern cardinal, and it produces a type of pigment called carotenoid, which specifically produces this red is astaxanthin. But what that carotenoid does, or what that pigment does, is you have sunlight. Sunlight produces all sorts of different colors. Most of those colors get absorbed by the carotenoid pigment in the wings. Or if it's melanin, it might be by melanin or some other pigment. And the only one here that is reflected is red. So that's why we see northern cardinals as red, is because of that carotenoid pigment absorbing all of these other wavelengths of light that the sun produces. So that's essentially how all pigments work, whether they are black, whether they're red, yellow, or other colors as well. The other variety that's a little bit less familiar to most people is structural color. Structural color involves interactions of light between materials of what we call different, I'm sorry, I'm losing myself, different indices, refractive indices. So what we have here is a non-iridescent color, structural color. What we have here is an iridescent structural color. And what this means is, let's say we have a bluebird. Bluebirds do not produce blue pigments. As far as we know, there are no birds out there, no matter what shade of blue they are, that produce a blue pigment. Blue pigment's very rare in animals. How they produces it instead is via this structural color mechanism. So what's going on here is we have a layer of keratin on the outside of the feather. We have a spongy layer of keratin on the inside that has a bunch of air spaces in between. And we have a layer of pigment down here. This is melanin specifically for this pigment. So what happens is you have sunlight coming in here, sunlight again producing all sorts of different colors. And most of those colors are just going through most of this structure and getting absorbed by the black melanin layer down here below. But what's really cool is that as this sunlight interacts with the air spaces and the keratin in the central layer here, it causes blue to bounce off. So the pigment itself is actually black, but the bird's feathers reflect blue because of this weird spongy layer in between. So this is like if you found a bluebird feather outside and you picked it up and held it up to the sun so that the sun was directly behind it, it no longer looked blue, it looked black because of this melanin layer. So this is essentially how non-iridescent structural colors work. Iridescent structural colors, similar kind of idea. The difference here is we don't really have those air spaces. We have kind of this weird crystalline structure of the melanin layers. So this gray layer here is keratin, followed by a melanin layer, followed by keratin, followed by melanin layer, followed by keratin. And what an iridescent color is like if you look at a hummingbird, and I have a video of this later on, if you look at a hummingbird and its head is turned one way, it might appear black or it might appear green. And if its head is turned another way, it might appear red or it might appear pink. So depending on your angle to that animal or its angle to you, it might appear a different color than if it's facing another direction. So the color changes depending on your viewing angle. And that's because of this structure here. So depending again on your angle, it might, light might be reflected back to your eyeball and it might appear purple or it might appear blue or it might appear teal, just depending on what angle you are to the bird itself. So these are structural colors. Pigment colors just again absorb light and reflect certain wavelengths, whereas we've got some weird interactions in the feathers of birds here that are producing these non-iridescent and iridescent structural colors. Which brings me to the most abundant of the pigments that contribute not just to pigmentary colors, but also to structural colors, at least for you melanin. So melanins are the most common natural pigments in the modern world. You have melanin in your hair. You have melanin in your liver. You have melanin in your skin. There's melanin in your cat. So for example, I like to use, this is my 15-year-old cat, Adria. She's calico, so she's got all the different types of melanins in her fur. What's going on here is she has a type of melanin called eumelanin that's reflecting black all over her fur on her body, and another type of melanin called pheomelanin that's reflecting red, or this kind of rusty red sort of color around her eye here or along her back here. So those are essentially the two types of melanin. Eumelanin is more common than pheomelanin is. If your hair is brown or if your hair is black, that's going to be produced by eumelanin. If your hair is naturally red, that's produced by mainly pheomelanin. So two different types of melanins. I should also mention pheomelanin can produce certain dull yellow colors as well. Not necessarily the colors of the goldfinch, but a little more dull than that. She also has white on her paws and on her face here. White is just the lack of pigment. So when light hits her fur on her foot, let's say, there's no pigment there to absorb any wavelengths or reflect any wavelengths. So all of the wavelengths are reflected. No pigment to selectively absorb certain colors like she has on the rest of her body. So white is the absence of pigment for animals. So pheomelanin and eumelanin. We also, in our hair at least, and in our eyes, our liver, for other animals, you can find these things also in hairs. You can find them in skin, not really in humans, but you can find them in feathers, etc. These are structures called melanosomes. What melanosomes are organelles inside of your cells that actually produce those two different types of melanins, pheomelanin and eumelanin. So this is a mud puppy. What I did here was just sample the skin of the mud puppy to see what the melanosomes looked like under a scanning electron microscope. This is part of my PhD research. And all of the little melanosomes here are kind of meatball shaped. That's legit, that is in the literature, meatball shaped. So very small, nearly spherical melanosomes. And again, melanosomes are going to be the organelles that produce melanin, both types of melanin. What's really cool is that in birds and mammals, you have different shapes and sizes of these melanosomes. So they are correlated to the color of that hair or the feather from the birds. In this case, what we have here, we have a rusty red colored feather, which you would expect to have more pheomelanin in it, producing that rusty red color. the melanosomes in rusty red colored feathers are meatball shaped and produce a lot of pheomelanin. So they are typically kind of a meatball shape. If you have black feathers, like in a cormorant, for example, they're more elongate, or what we call pill-shaped type melanosomes. So they're quite a bit longer than we see in the little meatball-shaped brown, red-brown type melanosomes. You have a gray feather. Gray feathers are kind of weird. Their melanosomes are longer and fatter than you actually see in the black feathers. And then what's really, really weird is when we get to the iridescent feathers. So iridescence, despite being a structurally produced color, is kind of weird because we have this weird crystalline structure of the melanosomes, but they're also really, really long, much longer than we find in black or gray type feathers, and they are arranged end to end in this kind of sheet-like orientation here. So this is helping to produce that iridescent structural color that you see in duck feathers or that you see in starling feathers and things like that. So very long. We have others as well, which one of my favorites are the platelet type melanosomes. So this is in the modern day, this is only occurring in hummingbirds. Hummingbirds have extremely highly iridescent feathers, especially the males. So this, for example, is Anna's hummingbird. You can find this guy out west. And as he turns his head, his feathers appear green on his head, but as he looks towards you, his feathers appear kind of pinkish purplish. And that is in part thanks to their crazy looking melanosomes. So this thing here is actually a not that thing. These things here are actually melanosomes, and they're kind of flattened, they're long, they're what we call platelet type melanosomes. I guess if you're familiar with pasta types, they kind of look like orzo. I don't know, I can't think of any other better way to describe them. But they are highly, highly reflective, and they produce these beautiful colors in hummingbirds. They also exist in ruby-throated hummingbirds, which are what we have around here in Pennsylvania. So these are super saturated iridescent colors, very, very highly iridescent in the hummingbirds. And again, currently hummingbirds are the only animals that have this type of melanosome. In this case, they're also hollow. Being hollow adds a whole other layer that I'm not going to go into, but that's kind of cool as well. So that brings me to talking about color in the fossil record. So since scientists first started studying dinosaurs in the early 1800s, we never really thought that we'd be able to actually determine what color the animals were. And this isn't just limited to dinosaurs, this is the whole fossil record wise. So typically when you see old art within or like 30 years plus or 25, even 20 years plus art of dinosaurs, for example, like Rudy Zallinger here or Charles Knight, even Jurassic Park, they are typically dull type colors, real drab, real boring, maybe matched to the area around them, the environment around them, etc. So the thought behind dinosaurs originally was that they were kind of dull, drab colors, kind of boring. And it turns out that is not actually the case. They are actually quite fascinating. So how do we know? I'm getting to that. Since the invention of the scanning electron microscope, which is what I use for a lot of my research, it's not really a light microscope like you're typically used to. It shoots electrons at your sample and allows you to see really, really tiny things like they're 3 dimensions or kind of in 3 dimensions. So these are scanning electron microscope images of these weird long bodies, kind of surrounded by this stringy stuff. This is from a paper, very famous paper called from Davis and Briggs, 1995, called Fossilization of Feathers. And when they looked at fossil feathers under the scanning electron microscope, they found these things and thought they were probably just decay bacteria that were surrounding the bodies as they fossilized and ended up fossilizing with the feathers themselves. So kind of cool, but at the time just written off as bacteria. Then in 2008, one of Derek Briggs's students at Yale, Jakob Winter, looked at some fossils from the Messel Pit in Germany, which is a little bit younger than, Crato Formation. Not the fossil pit in Germany, I apologize. But anyway, so he looked at some feathers, found those weird microbodies using scanning electron microscope in the feathers, and the bacteria explanation didn't really sit well with him. And he actually reinterpreted these as melanosomes. And one of the reasons for that is because you can see this feather is striped, probably striped in life. You've got some areas here that are kind of whitish where you don't really see the feather. And you've got some darker areas where you can see lots of detail on the feather. So he took samples from each of these areas and found these little microbodies in the dark areas, no little microbodies in the white areas. And okay, if they're bacteria, you're going to expect the entire thing to be covered in bacteria. And that wasn't the case. So we now have several lines of evidence suggesting that these aren't bacteria, including from one of the original authors himself, and that they were in fact fossilized melanosomes. So this paper was kind of the thing that shoved it all out there that we could actually potentially color dinosaurs and other animals. So his discovery, Jakob Winter's discovery, led to an explosion in dinosaur color studies. Starting off, one of the papers anyway, starting off is this one on Anchiornis huxleyi. So what they thought here, and this is, I have citations down below, what Lee et al. 2010 thought here was, okay, if we have melanosomes preserved in the fossil record, maybe we can see the same kind of size and shape correlation that we have with color that we see in modern day birds in some of these dinosaurs as well. And it turns out that is the case. So Anchiornis is a dinosaur from China. We've got the skull here. I'll just kind of orient you. The skull here and the body here. The skull is unfortunately separated from the body. And both the skull and the body are surrounded by these beautiful black feathers. So what this study did was they took many, many samples from throughout the animal's body, ran them under a scanning electron microscope, and found those same kind of microbodies in different preservational styles. Here's some 3D style, here's some impressions. And they compared those to the size and shape of those little microbodies to microbodies from a slew of modern day bird feathers. And they found that the size and shape of the microbodies in Anchiornis's feathers match up with the size and shape of melanosomes in modern day bird feathers. And they were actually able to do some statistics to determine that Anchiornis probably looked something like this. So you have around the crown feathers, you have these little tiny meatball-shaped melanosomes that suggest that the animal had a lot of pheomelanin in those crown feathers, making them red-brown colored. You have a lot of black or pill-shaped melanosomes in spots on the feathers, on the leg feathers here, a little bit here as well. So those are probably black. And then a lot of gray type melanosomes, those fatter pill-shaped melanosomes throughout the rest of the body. So where we have white here, they actually didn't find melanosomes. So they think that these areas of the leg feathers and the wings here were white. So this is one of the first color reconstructions of a dinosaur. And of course, this is going to lead to even more interesting fossil studies as well. This is from the same group, Lee et al. 2012, so 2 years after Anchiornis. One of my favorite dinosaurs is Microraptor. So Microraptor is another Chinese dinosaur. It had this beautiful body fossil right here, skulls over here, tails down here, and you've got feather preservation all along the body. They did the same kind of thing and ended up finding very, very long end-to-end oriented melanosomes, which suggests that this animal was iridescent in life. So Lee et al. 2012 for Microraptor was the first evidence of iridescence in the fossil record. Sorry, I'm reading the comments. Melanosomes were all found in fossilized rock of the dinosaurs. Yes, these are all of these melanosomes are from the feathers around the animal. What I don't mention or didn't mention here and don't have pictures of is we also take samples from the rock matrix around the fossil, and we have never found melanosomes in the rock matrix. It is just isolated to the feathers preserved here. So good question. So this was the first evidence of iridescence in the fossil record. Oops. But wait, there's something cool coming up here. This is Kaihong Jujai. So Kaihong, another Chinese dinosaur. This is Hu et al. 2018. And Orianchu, this is part and counterpart. We've got the skull up here. Here's the back, one of the wings, tails up here, legs are down here. So another really cool fossil from China that is surrounded by feathers. And same kind of idea. They looked at different samples of the feathers under scanning electron microscope, and all of a sudden they find these amazing platelet type melanosomes, just like what we see in hummingbirds. And again, these are only in hummingbirds today. So this is a huge surprise to find platelet type melanosomes. So what you're looking at here, this top row is all from Kaihong, the dinosaur. Bottom row is modern day different types of hummingbird melanosomes, platelet type hummingbird melanosomes here. So very similar in size and shape to the weird platelet type hummingbird melanosomes that produce that beautiful bright iridescence in modern day hummingbirds. Totally unexpected. So Kaihong, I'm sorry, this is going to be a little bright. Kaihong probably looked something like this. So you've got these crazy hummingbird type, platelet type melanosomes up here on the animal's neck and crown, along the top of the wings here, and then most of the rest of the body was covered in black pill-shaped melanosomes. So kind of a strange thing going on here that we would find hummingbird type melanosomes in this animal, and that's something that's still kind of being figured out. So anyway, Kaihong, one of my favorite examples, always a very strange one. So there's several other examples. I just don't have time to go through them. I'll go through one of mine, but we know that melanosomes can be preserved in fossil feathers. Okay, that's nice, really cool, amazing. What about the actual pigments themselves? What about eumelanin? What about pheomelanin? And that's kind of where a lot of my PhD research came in, was to look for these pigments in the fossil record. So there have been a number of techniques, different chemical techniques, some of which are very complicated, some of which are less complicated, to try to answer this question. Is eumelanin, is pheomelanin preserved in the fossil record? The most common of which, and the most accepted of which, unfortunately, is a method called TOF-SIMS, or time of flight secondary ion mass spectroscopy, which sounds like a mouthful. It is a mouthful. I have done some TOF-SIMS. It is a very difficult technique, but it gets you the results. So this is a paper, Lindgren et al. 2015, where they looked at fossilized melanosomes in a bird's eye here. So these are the melanosomes in the eye. This is the actual preserved eye here. And they determined that the fossil microbodies inside the eye, these little melanosomes, did have eumelanin in them based off of TOF-SIMS. So one example of a chemical technique that is used. The problem with TOF sims is it's very expensive, or quite often very expensive, and again, very difficult to interpret. So what I tried to do was a different technique, and I love this guy. This is my favorite fossil that I have ever worked on, and unfortunately, its name is CUGB P1202. We don't know what type of bird this is, but this is a 123 million year old bird from China that has kind of nice skeletal preservation. Most of the bones are there. But the cool thing is it's got these beautiful black feathers, including two elongate tail feathers and wing feathers surrounding the entire body. So this is a type of bird called an ananeornithine. They're also called opposite birds for reasons that I won't go into at the moment, but they're a completely extinct group of birds that lived along with the dinosaurs as well as the modern type of birds, the Neornithes. So due to its small size, it's much smaller than other animals within a group that we call the Bohyrnithidae. It's very small in comparison to other Bohyrnithids. It has pitted long bones, which means that it wasn't done growing when it died. We think that this was a subadult animal. So it wasn't a baby, but it wasn't an adult yet. And that'll come into play in a moment. The feathers suggest that it was probably male. The hypothesis here is that these two elongate tail feathers were used kind of like a peacock uses its tail, or a pin-tailed whydah, or some of those other crazy tail feathers that you see in modern day birds. So males usually have the ornamental feathers in modern day birds. So probably a male animal. We don't have other organ type evidence for that, but we're just going to go with it for now. That was your joke for today. So I took samples of this guy's feathers, and we find these amazing melanosomes. So we've got some 3D ones here. We've got some impressions here. We honestly don't know what's going on here, something weird. But these three samples at least had melanosomes. And what's cool is they're very, very long, and they're oriented end to end in kind of a sheet-like orientation, suggesting that this animal was iridescent in life. So this is my own drawing. I based it off of grackles. So iridescent in life, we don't know what color exactly it was, but at least weakly iridescent. So kind of cool. We also took samples to look at the chemistry, and this was the first paper to use a technique called Raman spectroscopy to determine if melanin was preserved in the fossil itself. Raman is nice because it's a lot cheaper and a lot easier to use than TOF sims and some of the other techniques. There's a couple of issues with it that I won't go heavily into, but what we've got here, Raman shoots a laser down at your sample, excites the molecules on the surface of the sample, and then essentially reads that excitement. So up top here on this readout, we've got modern day eumelanin from modern feathers. And down here we have the same kind of cool thing going on with our fossil feathers. So this graph is the fossils, this graph is the modern feathers. We've got two peaks, one peak here around 1360 and one peak here around 1570-ish. So we think we have eumelanin specifically preserved in this animal. We have tried pheomelanin with Raman. It does not work, unfortunately. So we can only use Raman spectroscopy to detect eumelanin. So we've got melanin in this fossil, which is really cool. So eumelanin does preserve in the fossil record. That's because eumelanin is a complex polymer that is very difficult to break down. Pheomelanin, a little bit iffy on that. Pheomelanin is something that is still trying to be figured out. It's not easy to detect using a lot of chemical methods, but there are a couple of papers that think they have it. So this is a really cool dinosaur called Borealopelta. It's an animal called a nodosaur, which is related to things like Ankylosaurus. It just doesn't, whoops, just doesn't have the big tail club that Ankylosaurus has. So this guy was found in Canada, and it has really amazing preservation. If you ever get the chance, go up to the Royal Tyrrell Museum in Alberta and see it. But Brown et al. 2017 looked at skin preserved on this animal, and they found possibly pheomelanin using some other chemical techniques. It's a little bit questionable, I'm not going to lie, because it deals a lot with sulfur, and sulfur is relatively common in rocks. Anyway, there's another study, Slater et al. 2023, that actually did find biomarkers for pheomelanin, suggesting that at least certain chemicals associated with pheomelanin can survive in the fossil record. So she found these in a bird called Confuciusornis, another dinosaur called Sinornithosaurus, and a fossil frog as well. So we've got maybe some evidence of pheomelanin. Pheomelanin still needs to be studied a little bit more heavily to determine what bits of it can actually survive in the fossil record, if anything at all. So we can find melanin in the fossil record. We can find melanosomes in the fossil record. What about the functions of these of the color in these fossils? Could extinct animals have used their colors for similar functions as modern day animals? Well, we don't really have any reason to assume otherwise. So here's what this means for the animals we've discussed. Anchiornis. Anchiornis has these beautiful, what we call spangles on its feathers. It's got some cute little freckles, I guess, on feathers on its face as well as its crown. These are very similar color patterns to what we see in nightjars, for example. Modern day nightjars. Nightjars use their colors for camouflage, so it's very possible that Anchiornis may have used its colors for camouflage as well. But typically when I see like this beautiful red-brown sort of crown feathers and these cute little freckles here, I'm thinking more sexual selection. So these may have also been used to attract females or to attract mates. So that explanation for these colors can't really be ruled out either. So maybe camouflage, maybe mate attraction. For the iridescent animals, iridescence in modern day birds is almost always associated with mate attraction. So iridescence is usually limited to the males in most bird species, if that species is iridescent. So for example, here's a male Anna's hummingbird with that beautiful pink sort of iridescent color. Here's the female, much more drab. That's not just limited to hummingbirds, that also occurs in many, many other types of birds as well. So chances are Microraptor and Kaihong, here's Microraptor, here's Kaihong, probably use their iridescent feathers to attract mates. Kaihong is also the oldest example of iridescence. So these traits are at least 161 million years old, which is really, really cool. Microraptor is a little bit younger. So probably mate attraction for that iridescence. Now, my favorite little guy, CUGB P1202. I forgot to mention the reason he doesn't have a name. He's a juvenile, or a subadult. Not a lot is actually known about his particular group of birds, so we couldn't really do any sort of analysis to figure out what type of bird he was, whether he's a young individual of a type of bird that is already named, or if he's a completely new type of bird. We couldn't determine that. That's why he's got the weird name. Anyway, so we know he's got iridescent feathers. We know he's got the elongate tail feathers. In modern day birds, those are typically associated with males. So those are typically male traits. He was probably a boy. So that's cool. Iridescence and the long tail feathers together also typically in modern day birds are used to attract mates. That's really cool. Here's the crazy thing though. In modern day birds, iridescence and ornamental feathers that are used to attract mates, like these long tail feathers, are typically only found in the adults. They don't develop in the juveniles or in the younger individuals. So we've got a young individual here with ornamental feathers, suggesting that these animals developed sexually before they were full-blown adults, unlike modern day birds, which are the opposite, where they develop into adults and then develop their ornamental mate attracting feathers. So this is not the first line of evidence for this. There's a lot of other evidence out there in the literature for this. It's just really cool that I got to work on a really cool example of this. So an interesting way that the opposite birds are opposite birds. Anyway, very cool animal. Next question, which dinosaurs can we color? The unfortunate thing is not a lot of them. So this paper, this is Lee et al. 2014. This paper hasn't come out yet. This is what I'm working on right now. Probably the last of my PhD papers to come out. So what this is going through all of the different types of vertebrates, looking at their melanosome size and shape. We've got modern day melanosome size and shape associated with color in these filled in little cartoons here. And then all of the hatched ones, whether they're colored or not, are fossils. So we don't have a lot of variation in melanosome size and shape in most of these types of animals. Really, that only occurs in mammals. So this is a cat. So it occurs in mammal hairs and it occurs in bird feathers. Very much so in bird feathers. Lots of variation in melanosome shape here. And it's only color coordinated in mammals and birds, not in these other modern groups. So if you look at the reptiles here, for example, you've got, they're almost the same shape for modern day melanosomes. And it's, for the most part, the same kind of idea with the fossils as well. Not a lot of variation. We can't determine the color of the lamprey samples that I took. We can't determine the color of the frog samples or the turtle samples or the lizards, etc. The weird one here is pterosaurs. We're figuring that out. Anyway, but if you go down the list here, here's a dinosaur, here's a dinosaur. All of these guys from here down are dinosaurs. And you've got tiny pill-shaped melanosomes in this animal, which is Psittacosaurus. It's related to Triceratops. You've got tiny pill-shaped melanosomes in this guy. This is Sinosauropteryx. These are its melanosomes. Not a lot of variation here. Where you start getting the variation is in the group called Pentaraptora. Those are the dinosaurs that are most closely related to modern day birds. So it includes things like the dromaeosaurs, which if Velociraptor were preserved with feathers, Velociraptor would have this type of variation. Microraptor, Anchiornis, they are all included in this group. CUGB P1202 would be down here. And then this is modern day birds. So we can only figure out the colors, except for maybe the pterosaurs, in Pentaraptoran dinosaurs, which includes modern day birds, and mammals, questionably pterosaurs. So we can't just dig up a T-Rex and maybe that T-Rex has feathers, maybe it has skin. We can't just dig up a T-Rex and determine what color it was, unfortunately. So it only works on certain dinosaurs. I'm sorry. Anyway, it sucks. So with that, let me check my time here. I'll go real quickly through some current ongoing research. I have started a lab, even though I'm only 2 years in, I have started a lab at Bloomsburg, and we're kind of shifting gears, still looking at color, but looking at mostly invertebrates. So we have seen colors in other fossils, not just dinosaurs, not just vertebrates. So we've got a really cool slab of crinoids here, for example. Some of these are white, some of these are gray, some of them are black. So some interesting thing is going on here with the colors of these particular animals on this single slab from O'Malley et al. 2013. We've got pink in crinoids from Vulcanstein here. So we don't just find weird color things going on in dinosaurs, we also find them in the invertebrate world as well. So one of the projects that I'm working on with a amateur paleontologist in North Carolina named Linda McCall is some cool invertebrates that washed up on North Topsail Beach in North Carolina that have some crazy looking colors. So these are about 30 million years old, and they have, this particular one is a sea biscuit called Hemiptegus carolinensis, and it's got purple in these little petalloid structures here, which is really, really cool. So we're trying to figure out, is that original color or is that something that came along later on? What's amazing about this site is that, and the reason why we have it, is that in 2015, there was an offshore dredging project to bring more sand up onto the beach. And that released a whole bunch of these crazy 30 million year old fossils. So thousands of fossils onto the beach collected by paleontologists and amateurs alike. And most of them have color patterns. So not just the Hemiptegus, which you can see that purple here again, and some spots of purples here as well, but also some of the clamshells. Barnacles have red on them. The clamshells have orange stripes. You can't see very well, but these brittle stars are pink. So we've got some cool color things going on here as well that I'm currently trying to do some chemistry work on and determine if actual original pigments survived the fossils here. These are some more examples. We want to know, do the North Topsail Beach fossils contain original pigments? What is making these spots on this crab, for instance, here? And what is making the purple here and the orange color here? We also want to know how they're preserved, things like that. What's interesting is we've also got some older fossils. This is Harduinia mortonus, which was recently dredged up 2023, I think, in Holden Beach, North Carolina. And these are 66 million years old. So they were lived along with the dinosaurs. So we've got some purple on these guys as well. This particular specimen, not so much, but we want to look at that as well. What we think is going on is that these animals were rapidly buried in a storm. They were rapidly dredged up during the dredging process in 2015 and 2022 in this case. And we just think that between those two factors, the color just happens to be preserved, which is really, really cool. Linda, the avocational paleontologist I'm working with, has also put some of these guys, specifically the Harduinia, out in sunlight, and she did notice that they do bleach over time. So very possibly we do have some pigments preserved here. We just have to find them. I also have a grad student named Henry Davies who's currently working on some fossil feathers that I'm really excited about. These are also Linda's samples. They're from the Creed Formation in Colorado. And one day, hopefully, I will have a student that can work on these cool insects also from the Creed Formation and determine what's going on here, because there's some cool color patterns going on in these insects as well. So stay tuned for that. Anyway, to kind of wrap up, why do we do these color studies? Why do we care about what color these animals were? Well, it gives us this really cool glimpse into the lives of animals that have been extinct for hundreds of millions of years that we never thought possible. We have found melanin dating back to over 300 million years ago, which is really amazing. And we never thought we'd be able to fill in the fossil coloring book and determine what color these animals were or how they interacted with each other based off of their color patterns, which modern day animals do all the time. So we're hoping to gain more insight into their anatomy, physiology, and relationships with each other and other types of animals based off of their preservation and their color patterns. We also hope to understand how long certain pigments last. Melanin and certain invertebrate pigments like quinones are the only ones that have been studied so far. So what about the carotenoids I mentioned before? What about other types of pigments? So which ones can last in the fossil record, which ones can't, which has some really cool implications as well. So anyway, hopefully I didn't go too long. Does anybody have any questions?
That was awesome. Thank you so
Thanks.
much. I know. So, first thing I gotta say is Jurassic Park got it wrong. When they were freezing because the big, large dinosaurs coming at them, they were like, "Oh my God, look at all those colors." They weren't like, "Oh, I'm gonna die." Like,
Yeah.
I do have to ask one question because this is something I learned from a scientist, from a PhD, actually only a couple of years ago. And it was at the Symposium for Friends of Mineralogy, PA, at Johnstown when we had it out in Johnstown. Iridescence, when you're looking, yeah, when you're looking at it under an SEM, you're looking for things that shift between like 5 and 10 nanometers or something like that. That's what creates iridescence. Is that something you know about?
You mean in terms of melanosome? Sorry, we're getting some feedback.
Yeah, Andy, can you mute real quick? Got some major feedback coming from you.
Sorry. You mean in terms of melanosome size, or do you mean just the difference in how light is reflected?
In minerals and in rocks, it's been proven that when you get an iridescent rock, like peacock ore and stuff like that, it's a fracturing on the microscopic level at about 5 to 10, I think, nanometers. Is that potentially the same size as your little pills that are lined up?
Oh, the melanosomes are bigger. So the little meatball shaped melanosomes that produce like the red brown colors, those are about 500 nanometers. All the other ones are over a micron. So they're over 1,000 nanometers in length. The iridescent ones,
Okay.
sorry?
Maybe it's a micron. I gotta.
That could be. I don't know about iridescent minerals. That's something that I definitely need to look into. But the super elongate melanosomes that we find in like CUGB P1202 or in Microraptor and some other dinosaurs are 7 microns long, which would be 7,000 nanometers. Some of them are even longer.
That
So it
makes
just depends.
sense. I think, yeah, yes. All right. So I think that's probably what I just couldn't remember the actual measurement unit. So
it could be the same kind of idea.
Yeah.
You've got crystals and minerals, you've got crystals, melanosomes making crystal shapes. So possibly.
Yeah, and it's just the way the light bounces off of it, really. That's all. And in crystals, it's just a fracturing. When you have fractures that are that close together, that's kind of what creates moonstone and some of those iridescent kind of crystals that we find out in nature. Very cool that it applies all together.
Cool.
Yeah, we gotta take a look at that. Does anyone look, did you looking through some of these, did you go to
Yeah, I see a couple of questions in the chat. So Deb B asks, did you go to China to study that bird? I did. I've been to China twice. Once on the way back from Mongolia for my undergrad research. And then in 2014, I went for my PhD research. And it was pretty awesome for the most part. But yeah, I met a lot of really cool fossils there and a lot of really cool Chinese paleontologists there as well. But CUGB P1202 is still in China. I don't know if it's on display or not. CUGB stands for China University of Geosciences. So Kwangguo Li, who is the main author on a lot of those papers I mentioned, he actually had that fossil in his office when I met him, and I was supposed to be working on something else. And I was like, I love this bird. Can I work on this bird? And he said yes. So yeah. That was my first paper too. My first author paper.
Very cool. Very cool.
So yeah, I also got a little bit kidnapped in China temporarily too. So yeah, there's that as well. But
Oh my gosh.
anyway, my advisor and I both. We went to the Forbidden City one of the last days that we were there, and the Forbidden City closed while we were there, and everybody was escorted out, of course. And I left my backpack in the women's restroom inside the Forbidden City. So we had to stay behind, ask the nice Chinese people, hey, can I get my backpack back? Thankfully, it was still there. They found it. But we were some of the last people out of the Forbidden City, and we were really frustrated, ended up on some motorcycles to get back to our hotel. There was our mistake. We got separated from each other, my advisor and I, on separate motorcycles, and they took us to one of the few parts of the city that didn't have any cameras and tried to get money out of us. From what I don't remember it very well, from what my advisor said, and I was a runner at the time and much more fit than I am now, I did some weird basketball dodge move around the guy that was trying to get money from me, and we just booked it. So temporarily kidnapped in China. Anyway. So yeah.
Wow.
But I did
a
like
story right
China.
there. That's awesome. That's awesome.
Yeah.
I think Debbie also asked, how did you get the sample off of the feather?
Yeah, this is something I just taught my grad student how to do a couple days ago. We have, I wish I had a picture of this, but we have needle probes that they're not quite as thin as needles are, not quite as sharp as needles are, but we use those to kind of bring up very tiny samples off of the feathers so that we don't destroy them. And those tiny little samples we can actually stick on SEM stubs and coat in metal to do SEM with. And we take separate samples from right next to those samples to do the chemistry stuff with. So they're very teeny tiny. They're usually only about 1 to 2 millimeters in length. And so far, most of the techniques that we use can work on fossil samples that small.
For those that have never used an SEM, this is in a Dirtman report that's currently unreleased that actually is sitting on my desktop on this computer right now that I am still trying to get through. Basically, an SEM works in a completely oxygen depleted environment, and it also cannot have any water. It cannot have a lot of stuff because it'll just get sucked out in the process of it preparing to take the images. So, organics have to be really dry. So, if you put one in, I don't think you can actually do organics in an SEM, can you?
You can, sort of. So it depends on what type of SEM you're using. If you're using an environmental SEM, they don't take as good a picture of images, but you can put organics in there just fine. For the type of SEM that we used, which is a field emission scanning electron microscope, it's all in a vacuum. It has to be dry. I actually took some samples from some eyeballs, modern day eyeballs. Not human, I'm not that crazy. And we had to fix them in a chemical called glutaraldehyde and then put them in plastic so that we could run them and they would be completely dry. So yeah, for most SEM samples, they have to be completely dry and fixed, but you can do some organics. There is actually
So,
there is one animal that we know of that can survive in the vacuum of an SEM. They can also survive in a vacuum of space, and that's the tardigrades, which are tiny little animals. They're also called water bears and moss piglets. But
right,
they can survive,
about
which is
those.
cool.
Yeah, yeah. So yeah, I just so everyone knows, over the course of Parksburg, I have collected samples from every color of soil out there. So I have, I don't know, 20 different samples of soil to eventually go to an SEM because I'd really like to know a lot more about Parksburg. We all know it just got shut down. I'm gonna hopefully get there this weekend just to film one last scene. I am filming a Dirt Man report that will finish once there's a paved parking lot and buildings over top of it. But I just so everyone knows, I am collecting samples that will get tested at some point, hopefully, under an SEM
Cool.
to find out what the pink soil was, what the purple was, what the white, what the black, what the, this crushed pegmatite zone, what we actually were looking at there. But I did have to, I have dehumidified them, all of the different things. So real quick, iPhone has said in the comments, bonus nanometer info. The light that's carrying this meeting along the fiber optic cable is traveling wavelength of 115. All right, you all can see that.
That's cool.
Yes, Deb, that's the cookies and cream site. I'm not worried about putting Parksburg out there at this point because it's closed. Everything's gonna get covered up in a matter of a week or two now. So, all right. Well, thank you, Dr. Jen Peteya. Let's do our drawing for the raffle. We've got the box saying fragile right here. I gotta take that big label off of there. But here we go. I'm going to share good old Google. Here we go again. So between a number of 1 and 100 in the comments, get it in now. You've got 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, here we go. Generating the number. 74 has won. So who was closest to 74 without going over? 65. Oops. We were at 74. So we have a 77. That's just over. 76 is just over. I am seeing 65.
65.
Yep. Jen Peteya. Dr. Jennifer Peteya, you won tonight's raffle. What a bonus. Thank you for coming. You win tonight. That's funny. Oh boy. All right, cool. Well, Jen, I will be contacting you for a mailing address, and I'll be sending this box to you on Thursday.
Thank you. That's awesome.
You're getting a nice good these are actually some pretty good specimens in here, so congratulations. All right, well, does anyone have anything they'd like to say, talk about? I do want to put it out there. I meant to say this. There are two things I do need to say real quick. One, if anyone has a Dirtman report like thing, if you all want to film your own thing that's 5 minutes or so ish, send it to us. I would love to totally foster a new Dirtman. I only started doing this because of Jerry Jones and the Rock Room. This is Geology Club. We're doing this on Zoom. Let's get a new Dirtman out there. So anyone out there who's seeing this, film some stuff. Give me a 5 minute something. I will totally review it. And if it's good. Let's show it. So, send me your stuff. Second announcement I'd like to make real quick before the end here. I am going to send out an email to everyone in the club. I am opening up signups for Mohawk Valley for the Labor Day weekend. So we have closed out Diamond Mountain. The free members, they have all been picked. They are all done. But Mohawk Valley Mineral Mining is extending this offer out to everyone in the club. So I'm going to send this back out to everyone. You are able to attend and sign up. I'm also going to invite Central PA Rock and Mineral Club. Let's get everyone out there. Let's share this one. I was happy as hell to talk to the owner and get that kind of a deal. The last deal I got with him was $60 per day for digging. So, and the Dirtman deal that I've always shown people, you can always get 10% off with just saying, me up there. But this is, this was a deal that I want to extend to everyone. So, the owner's all for getting as many people there as possible. So come on up. Everyone is invited to Mohawk Valley Mineral Mining, free camping in the field. I will send out details to everything now that I know them. Excuse me. And that's my announcements. So thank you, everyone. Does anyone have anything? I didn't see anything pop up in the comments. So on behalf of Jake, who's not here, running some awesome show somewhere in the country, Andy, do you have anything?
Real quick, I just want to thank Bill Geary for jumping in at Pickering when Jake was sick. Bill took the reins and sort of led the group at Pickering at the last minute, and really appreciate that. And like you said, we'd like to get some members to share what they're finding. If like you said, if it's a little video or whatever, yeah, we want to know what you're finding out there.
Hell yeah. And I have already messaged Bill, and it looks like he's on there on right now. So Bill, I've already messaged you. I want you to lead the Diamond Mountain trip since I'll be at Mohawk. You just have that rock breaking mentality. So anyway, if you all can still see me, I just got a notice saying my connection was unstable, although I'm on Wi-Fi. So anyway, thank you all for attending. This will go out on YouTube probably tomorrow because I want to send it to someone in California who's interested in seeing this. So first time this will be going out to California, I think. So thank you all. And oh, Bill was the iPhone. Okay, now we got it. All right, cool. All right, thank you all. And we will see you next month. I'm not sure who's going to be the presenter next month yet. It might be me, and I might be doing one on Diamond Mountain and Mohawk, which bringing you stuff no one's seen, except for Central PA. Central PA saw it a couple years ago, but I'm going to be bringing stuff that I haven't released. So, all right. Thank you all, and we'll see you next month.